Bacteria are classified based on their morphology or the shape. Herein, you can see the bacteria are in the form of spherical or oval shaped, and these are called as coccus. Coccus means berry. The second is the bacilli, which means it is rod shaped. The vibrios are comma shaped or they have a vibratory motility. Spirilla are bacteria which have a rigid spiral forms, that is just like a spiral shaped staircase. Spirochetes are flexible and spiral in shape. Mycoplasm do not have cell wall, therefore they do not have a stable morphology. There are other forms of bacteria such as the actinomycetes, which are branching filamentous bacteria. That is, they'll be having branches just like a tree. Actus meaning ray and myx meaning fungus. As it looks like the rays of the sun, therefore the name is actinomycetes. Again, the cocci bacteria can be classified as single-celled, diplococci, which occurs in pair, tetrad, which are group of four, sarcenia, which are cube-like shape, streptococci, which are chain-like morphology, and staphylococcus, which are grape-like clusters. Similarly, bacilli can also be again classified based on their various shape. Bacillus means single unattached cells that looks like a rod. Example are Bacillus cereus and Salmonella. Diplobacilli, these are two rods are attached to each other and are found in pairs after cell division. Example, we have Moraxilla and Klebsiella. Streptobacilli, due to cell division, in one plane, bacilli are arranged in a chain form. These are genus Streptobacillus contains gram-negative aerobic or facultative bacteria, and other examples as you can see here. Cocobacilli, these are short compared to the other bacilli and oval in shape. They appear like a coccus. Example, Chlamydia. And often these are confused for cocci bacteria. And the other type of bacillus is palisades. The bacillia after cell division bend and therefore are arranged as Placidae, that means fins-like structure. For example, Corynebacteria, diphtheria. Based on the staining technique, the bacteria can be classified as gram stain and acid fast stain. Gram stain was invented by Christiana Gram, and hence it is named as gram stain, and there are two types. If the bacteria retain the primary stain, that is, if they appear violet under the microscope, they are called as gram-positive bacteria. And if the bacteria take up the counter stain, that is the safranin, they appear red in color and are called as gram-negative bacteria. In acid-fast stain, the most commonly used is the Ziehl-Neelsen stain. Acid-fast bacteria appear red in color when they are seen under the microscope. Example, Mycobacteria tuberculosis. Again, based on their gaseous requirements, the bacteria are classified as obligate aerobes, those bacteria that grow only in the presence of oxygen and cannot grow in the absence of oxygen. Facultative anaerobes, those bacteria that are able to grow either with or without free oxygen. That is, we can say that even in the presence or even in the absence of oxygen, these bacteria can survive. Microaerophils grow best in the presence of low levels of oxygen than that present in the atmosphere. Obligate anaerobes, these bacteria that can grow only in absence of oxygen. In fact, these bacteria die if the oxygen is present. Capnophiles, those bacteria that require carbon dioxide for the growth, that is, they are carbon loving bacteria. Based on the flagella, the bacteria can be classified again as those which are motile and non-motile. Bacteria which possesses one or more flagella are called as motile bacteria. Non-motile bacteria are without flagella. Non-motile bacteria without flagella are called as atrichous bacteria. Based on the different type of flagella, herein you can see monotrichous bacteria means single flagella at one pole. Lophotrichous means tuft or flagella at one pole. Amphitrichous means flagella at both the poles. Peritrichous means flagella all over the body of the bacteria. Amphilopotrichous means tuft of flagella at both the ends. So, this amphilopotrichous is a combination of these two. Okay, that is, they have flagella on both the ends as amphitrichous and also at the they are in the form of tufts. Classification based on spore forming. Bacteria again are of two types based on the spore forming bacteria and non-spore forming bacteria. Those bacteria that produce spore during unfavorable condition are called as spore forming bacteria. There are of two types, endospore and exospore. Endospores, spore production within the bacterial cell wall, and exospores means spore produced outside the cell of the bacteria. Non-sporing bacteria, those bacteria which do not produce spore. Classification based on pH. Acidophils are those bacteria which grows best at the acidic pH. That is, it can be from 1 to 5. Alkali Alkaliphilus are those bacteria that grows best at the alkaline pH, that is around 8 to 12. Neutrophiles, those bacteria that grow best at the neutral temperature. Classification of bacteria based on the mode of nutrition, and this is one of the very important aspects. Phototropes are bacteria that gain energy from the sunlight. Chemotropes gain energy from chemical reactions. On the basis of the source of electrons, the bacteria are classified as photolithotropes. That is, these bacteria uses inorganic compound as electron source. Photoorganotropes uses organic compounds as electron source. Autotrophs are bacteria which uses carbon dioxide as a sole source of carbon to prepare its own food. Bases of energy utilized to assimilate carbon dioxide. These are of two types, photoautotrophs and chemoautotrophs. Photoautotrophs means they utilize light to assimilate carbon dioxide, and chemoautotrophs utilize chemical energy for assimilation of carbon dioxide. Basis of source of electron, we have photolithotrophic autotrophs and photoorganotrophic autotrophs. So coming to the basis of electron, that is the chemotropes, can be classified as chemolithotropes uses inorganic compounds as electron source, and chemoorganotropes uses organic compounds as the source of electron. Bacteria which are unable to grow with carbon dioxide, these bacteria are dependent on preformed organic compounds, and these are called as heterotrophs. So most of the human pathogenic bacteria are heterotrophs in nature. Based on the temperature requirements, temperature at which bacteria growth occurs best is known as the optimum growth temperature for the organism. The lowest temperature at which the organisms can survive and replicate is the minimum growth temperature. The highest temperature at which growth occurs is known as the maximum growth temperature. Based on this, we have psychrophils, that is cold loving. These bacteria grow at 0 degree centigrade, but the most optimum growth occurs at 15 degree centigrade. We have mesophiles that are grow at moderate temperature between 20 degrees centigrade and 45 degree, and with an optimum growth temperature range of 30 to 39 degrees centigrade. What that means that this is the range at which they can grow, but the best is 30 to 39 degrees centigrade. So most of the human pathogenic bacteria grow at 37 degrees centigrade. Organisms that grow optimum temperature at 50 degrees centigrade to a maximum of 80 degrees centigrade are called as thermophiles, that is heat loving. They do not multiply at room temperature and are usually found in hot springs. Hyperthermophils, these are extremely heat loving, has an optimum growth temperature above 80 degree centigrade. So based on the temperature, we have psychrophils, mesophils, and thermophils and hyperthermophils. Classification based on the presence of salt, they classified as halophiles, those bacteria that require sodium chloride for growth. The name halophiles come from the Greek word known as salt loving. Halotolerant do not require salt but can grow under the saline condition.